For more than 3 billion years, life on Earth remained microscopic. Oceans held bacteria and algae pressed into layered mats along the seafloor. Soft bodies absorbed nutrients from the water. Nothing chased, nothing fled. There were no eyes, no skeletons, no predators. Then, roughly 541 million years ago, the fossil record changes. Within about 20 million years, hard shells begin appearing across distant coastlines. Sediments that once preserved only impressions now hold mineralized armor. Bodies gain direction, front and back, inside and outside, mouths open, guts form, muscles attached to structured frames. Fossils show burrows cutting through ancient seabeds for the first time. Other fossils carry marks that can only be explained as damage from feeding. Nearly every major animal body plan alive today traces back to this window. Trilobites, early chordates, primitive arthropods, the structural blueprint of fish, reptiles, mammals, and eventually humans begins here. But why did life remain simple for billions of years before this transformation? What shifted in Earth's oceans that allowed complexity to stabilize? How did genetic systems begin building coordinated bodies rather than flat sheets of cells? And why did this transition occur so abruptly in geological terms? Tonight, we follow the most important 20 million years in Earth's history. But before we begin, if you enjoy journeys like this, soothing and quietly filled with wonder, I'd love it if you gave this video a like and maybe tap subscribe. And if you feel comfortable sharing, I'd love to know in the comments, where are you tuning in from tonight and what time is it there? It's always a joy to see how far across the world these quiet stories travel. Now take a deep breath, let your shoulders soften, and together, let's trace the moment when the first animals began reshaping the planet. Long before shells pressed into stone or mussels carved pathways through sediment, the planet carried a different rhythm, broad and unhurried, like a tide that never quite turned. 600 million years ago, continents lay low beneath warm, shallow seas that spread across their surfaces in luminous sheets. Coastlines were wide and intricate, and sunlight filtered easily through clear water, touching the seafloor across immense distances. There were no reefs built from skeletons, no forests of branching animals swaying in currents. The ocean floor was covered instead by living carpets, dense microbial mats layered in films of bacteria and algae, each sheet bound together by secreted threads and fine grains of sediment. These mats formed textured plains that stretched farther than any modern meadow. They trapped particles drifting down from above and held them in place, stabilizing the boundary between water and stone. When storms passed overhead, the disturbance rarely penetrated deeply. The surface healed, the layers persisted. In these quiet expanses, chemistry unfolded gradually. Oxygen, released by photosynthetic microbes, accumulated in the water column and seeped into the atmosphere. Its concentration was rising compared with far earlier ages, yet it had not reached the richness familiar today. The balance remained delicate, sufficient for simple multicellular bodies, but not yet abundant enough to support large, energetically demanding forms. Across this subdued seafloor lived the organisms of the Ediacaran world, soft-bodied and often quilted in appearance, their forms resembling fronds, discs or elongated cushions pressed gently against the substrate. Many absorbed nutrients directly through their outer surfaces, drawing sustenance from surrounding water without mouths or internal digestive tracts. Their tissues left only faint impressions when buried, subtle outlines that spoke of symmetry without skeleton. There were no jaws to bite into flesh, no mineralized armor to resist attack. Hard parts had not yet entered the design vocabulary of life. Ecological interactions were correspondingly restrained. Without clear evidence of active predation, competition unfolded slowly, shaped more by space and chemical gradients than by pursuit or escape. Communities changed, but at a pace that allowed continuity. Sediment remained largely undisturbed, its layers lying flat and intact beneath the microbial cover. Nutrients cycled within stable boundaries. Energy flowed, yet it did not surge. This was a biosphere that functioned, expanded, and endured, but it did so without urgency. Complexity in form was limited, and behavior left little trace. The foundations of later transformation were present in chemistry and in genes, waiting within cells that divided and differentiated in simple patterns. For millions upon millions of years, the ocean held this steady state, a world complete in its own logic, poised quietly on the edge of change that had not yet declared itself. In stone that once lay beneath shallow seas, there are moments where the record tightens into something unmistakable. A curved fracture along the rim of a shell, a circular perforation drilled with patient precision, a pattern of damage that repeats across distant outcrops, separated now by oceans but once joined beneath the same water. These marks are not accidents of pressure or burial. They follow the geometry of mouths, the leverage of grasping limbs, the controlled force of an organism pressing against another living body. For vast stretches of earlier time, such traces are absent. Impressions linger softly in sediment, but they do not carry the signature of violence. Then, within Cambrian layers, the surfaces change. Trilobite carapaces bear healed injuries, crescents of regrown mineral where a predator's attempt failed. Small shelly fossils show punctures aligned with emerging mouthparts. In some specimens, the hole narrows inward, tapering as if a rotating structure bored steadily through calcium carbonate. The interaction is recorded at the scale of millimeters, yet its implications reshape entire ecosystems. Once feeding becomes active rather than passive, anatomy responds. Organisms that had absorbed nutrients directly from surrounding water begin enclosing their tissues within protective boundaries. Calcium carbonate and calcium phosphate are secreted in thicker layers, forming armor that can resist crushing or delay penetration. Over successive strata, shell walls increase in density, their microstructure reorganized into interlocking crystals that distribute stress. Edges become reinforced, apertures narrow. Other lineages choose a different strategy. Spines project outward from soft bodies, first as modest protrusions, later as elaborate arrays that complicate a predator's grip. Some extend like needles, others broaden into plates that overlap, creating articulated defenses. The fossil record preserves these modifications not as isolated curiosities, but as trends, each generation slightly more fortified than the last. Selection operates quietly, yet persistently, filtering designs through survival. Predators change as well. Appendages capable of grasping evolve greater curvature and strength. Mouth parts develop serrations, ridges suited for scraping or tearing. Sensory structures orient forward, aligning perception with pursuit. Burrows in sediment grow more complex, indicating movement through layers once left undisturbed. The seafloor becomes a terrain of concealment and exposure, where hiding and hunting leave intertwined signatures. This is the beginning of an evolutionary dialogue carried in mineral and muscle. Every injury recorded in stone implies an encounter. Every healed scar suggests resilience. Energy flows differently through such systems, redirected into speed, reinforcement, detection, and evasion. Anatomy ceases to be merely a way of existing within water and becomes a negotiation with other bodies sharing the same space. From this point onward, survival does not rest solely on chemistry or position. It is shaped by interaction, by the constant refinement of structure in response to pressure applied by another living form. The marks in ancient shells are small, yet they announce a profound shift, the first clear evidence that life had begun to consume life in ways that would echo through all subsequent oceans. Light had always poured into the ocean from above, scattering into blue gradients and fading with depth, but for ages it touched bodies that could not interpret it. It warmed surfaces, drove photosynthesis, and traced gentle patterns across sediment, yet it carried no image. Then, within Cambrian strata, mineral surfaces appear that suggest something new, arrays of tiny lenses arranged in precise curvature, each one angled to gather a fragment of the world beyond the body that bore it. Trilobites preserve these structures with unusual clarity. Their compound eyes, built from calcite, remain fossilized as mosaics of hexagonal facets, each lens once focusing light onto photoreceptive cells beneath. Unlike modern arthropods that rely on organic material alone, these early forms used crystalline mineral to shape incoming rays. Calcite, transparent and abundant in Cambrian seas, became part of anatomy. Its optical properties were not incidental. The crystal lattice guided light efficiently, reducing distortion and enabling sharp detection in the shifting brightness of shallow waters. The geometry of these eyes reveals more than appearance. Facet, size, and curvature vary between species, implying differences in resolution and sensitivity. Some possessed broad fields of view suited to scanning horizons, others more restricted arcs that favored forward orientation. Such variation suggests that vision was already being refined by ecological context. To see is not merely to register brightness, but to detect motion, contrast, and direction. With visual input, an organism can align its body toward opportunity or away from threat before contact occurs. Behavior shifts accordingly. Locomotion becomes purposeful rather than exploratory. Appendages coordinate with what lies ahead rather than reacting solely to chemical gradients or touch. Predators equipped with forward-facing visual arrays can track silhouettes against sunlit water, adjusting trajectory in response to subtle changes. Prey species respond in turn, developing spines that disrupt outlines or adopting body shapes that blend with sediment and shadow. The interaction between hunter and hunted gains a new dimension, mediated by photons traveling through clear seas. Neural tissue must adapt as well. Photoreceptors connect to processing centers that integrate multiple inputs into coherent perception. Even in these early animals, genetic pathways guiding eye development involve regulatory networks that orchestrate symmetry and segmentation. The emergence of eyes, therefore, reflects not a single structural novelty, but coordination between optics, nerves, and movement. Light becomes information, and information reshapes survival. Sediments record this transformation indirectly. Trackways grow more directed, less meandering. The distribution of burrows changes as animals respond to visual cues above the seafloor. In communities where sight spreads, the tempo of interaction increases. Detection precedes encounter. Anticipation replaces randomness. Ecosystems begin to operate at a different pace, not louder or more chaotic, but more responsive. From this point onward, the ocean is no longer experienced as uniform brightness or darkness. It becomes a field of signals, patterns in motion, gradients of possibility traced through water by eyes that have learned to see. There are impressions in fine-grained shale, where the outline of a body curves like a drifting ribbon, segmented and supple, preserved in detail, so delicate that even the softest tissues remain visible. In these stone pages, from what is now called the Burgess Shale, one form appears again and again, larger than its contemporaries, built not for stillness, but for motion. Anomalocaris moved through Cambrian water with a flexibility unlike the armored crawlers below, its body composed of lateral lobes that rippled in sequence, generating lift and forward thrust in a smooth, coordinated wave. At the front of this animal extended a pair of articulated appendages, jointed and lined with spines that curved inward like the tines of a careful instrument. These frontal grasping limbs were not rigid spears but responsive structures, capable of bending and closing around struggling prey. They framed a circular mouth positioned beneath the head, composed of overlapping plates, arranged in a radial pattern. When contracted, those plates formed a firm opening capable of exerting pressure sufficient to crack or tear softer tissues. The arrangement suggests repeated use, a design refined through encounters that demanded strength and precision. Unlike many creatures anchored to sediment or confined to short bursts of crawling, Anomalocaris occupied open water. Its swimming ability allowed it to patrol above the seafloor, scanning the shifting landscape with large, well-developed eyes set on short stalks. These visual organs, complex and multifaceted, provided wide fields of view and sensitivity to movement. In clear Cambrian seas, such perception would have revealed silhouettes against filtered light, enabling pursuit before contact. The coordination between sight, propulsion, and grasping limbs implies neural integration that supported deliberate hunting strategies rather than accidental collision. Evidence of these interactions lies within the same deposits. Some trilobite fossils from Cambrian strata display injuries consistent with powerful compression, their carapaces bent or fractured in patterns that match the circular mouth parts attributed to Anomalocaris. Other specimens show healed damage, suggesting that not every encounter ended in success. The predator and its prey were engaged in a dynamic exchange, each adaptation met by another. The presence of large, actively swimming carnivores marks a further escalation in ecological structure, where size and mobility conferred advantage. Within the Burgess Shale assemblage, Anomalocaris stands out not merely for its dimensions but for its combination of traits. It integrated vision, speed, and specialized feeding apparatus into a single body plan, occupying a role near the top of its food web. Such an organism would have influenced population distributions, driving smaller animals toward concealment, thicker armor, or altered behavior. Energy pathways through the ecosystem shifted accordingly, channeled through the musculature of a predator that ranged widely rather than remaining fixed. In this Cambrian ocean, the emergence of an apex hunter signals that ecological networks had matured into layered hierarchies. The water column itself became structured by pursuit and avoidance, by the silent passage of a form whose anatomy reflects both opportunity and pressure. Through shale and time, its imprint endures, a testament to how swiftly life organized itself into complexity once the tools for seeing, grasping, and swimming converged within a single, formidable frame. Some Cambrian bodies seem less like ancestors and more like sketches from a restless imagination, as though evolution were testing possibilities without yet settling into familiar outlines. In fine-grained deposits where soft tissues were captured before decay could erase them, silhouettes emerge that challenge expectation. Their symmetry is present, yet their proportions feel unfamiliar. Their appendages arranged in ways that do not map neatly onto modern phyla. Hallucigenia appears first as a narrow tube balanced upon pairs of slender limbs, its back lined with elongated spines that project upward like a defensive forest. For years, its orientation puzzled paleontologists, the creature reconstructed upside down before closer study revealed which surface met the seafloor and which faced open water. The spines likely served as deterrents, rigid structures that made swallowing difficult for predators refining their tactics. Beneath them, soft legs ended in delicate claws suited for grasping microbial substrates or clinging to uneven terrain. Its anatomy reflects both vulnerability and innovation, a body experimenting with protection without sacrificing mobility. Opabinia presents an entirely different arrangement. 5 eyes crown its head, spaced across a broad frontal region, offering overlapping fields of perception. Extending forward from beneath those eyes is a flexible proboscis, jointed and tipped with a grasping structure capable of transferring food toward a rear-facing mouth. The combination appears almost improbable, yet in Cambrian seas it functioned within a living network of pursuit and evasion. Multiple visual inputs would have enhanced detection of movement, while the proboscis allowed precise manipulation of small prey items or organic fragments suspended in water. Wewaxia, armored yet soft beneath, carried rows of overlapping sclerites across its dorsal surface, each plate composed of mineralized tissue embedded within an organic matrix. Long spines projected laterally, adding another layer of defense. Its underside bore a rasping organ, comparable in function to a radula, enabling it to graze along surfaces with controlled motion. Here, protection and feeding apparatus are integrated into a compact form, suggesting that ecological roles were already diversifying beyond simple categories of hunter and hunted. These organisms illustrate a phase of evolutionary exploration in which developmental pathways generated a wide spectrum of body architectures. Genetic regulatory systems, still plastic and responsive, permitted variations in segmentation, appendage placement, and sensory distribution. Not all designs endured. Many lineages documented in Cambrian strata leave no direct descendants in later periods. Their absence in subsequent rocks does not diminish their significance. Rather, it underscores the experimental character of early animal evolution. In this interval, life was not constrained by the later stability of established phyla. Structures could appear, persist for millions of years, and then vanish as environmental pressures shifted and competition intensified. The fossil record preserves these forms as reminders that the path toward modern ecosystems was neither linear nor predetermined. Within Cambrian waters, anatomy unfolded across a spectrum of possibilities, some of which would echo forward through deep time, while others would remain confined to this singular, inventive chapter of Earth's history. In certain Cambrian layers, the sediment changes character, no longer yielding only impressions and soft outlines, but fragments that glint faintly within the rock, tiny cones, tubes, plates, and caps no larger than grains of sand. These remains, collectively known as small shelly fossils, appear across widely separated regions, embedded in limestone and shale from what were once distant continental margins. Their distribution suggests that a profound biological shift was unfolding not in isolation, but on a global scale. Each fragment represents tissue that had learned to incorporate minerals drawn from seawater. Calcium carbonate and calcium phosphate, dissolved within the Cambrian Ocean, were precipitated by living cells into organized structures, forming shells, spicules, and protective coverings. The chemistry of the water made such mineralization possible, yet the decision to use it belonged to evolving genetic programs that regulated when and where crystals formed. Microscopic control over nucleation sites allowed organisms to shape rigid elements with remarkable precision, arranging crystals into layered microstructures that balance strength and weight. Hard parts altered survival in immediate and lasting ways. A mineralized shell provided resistance against compression and tearing, shielding vulnerable tissues from grasping limbs and crushing mouthparts that had become increasingly effective. Even thin skeletal elements could deflect a probing appendage or delay penetration long enough for escape. At the same time, rigid frameworks offered internal support, enabling muscles to attach more efficiently and bodies to grow larger without collapsing under their own mass. Protection and structural integrity became intertwined advantages. As more lineages adopted mineralized coverings, interactions intensified. Predators capable of breaching softer prey now encountered resistance, prompting refinements in feeding strategies. Crushing surfaces thickened, biting edges sharpened, and behaviors adjusted to exploit weaknesses in shell design. In response, shells themselves diversified. Some thickened, others developed ridges, spines, or complex ornamentation that distributed stress and discouraged attack. This reciprocal dynamic is often described as an arms race, though its tempo remained measured across geological time, unfolding through countless generations of incremental change. The fossil record reflects the rapid proliferation of these innovations. Small shelly fossils appear within a relatively narrow stratigraphic interval, marking a horizon where mineralized remains become abundant and varied. Their presence across continents indicates that similar solutions were emerging in parallel, shaped by shared environmental conditions and ecological pressures. What had once been rare becomes common, and the seafloor, previously dominated by soft bodies, acquires a new texture composed of durable fragments. With skeletons came durability in the geological sense as well. Hard parts increase the likelihood of preservation, amplifying the visibility of animal life in stone. The rock record grows denser, more detailed, as mineralized tissues resist decay and compaction. Through these scattered shards and intact shells, it becomes clear that the architecture of life had entered a new phase, one in which chemistry, genetics, and interaction converged to produce bodies capable of enduring both predation and deep time. Beneath the mineral shells and articulated limbs, beneath the spines and eyes that now filled Cambrian seas, a quieter architecture was taking shape, written not in stone, but in sequences of nucleotides coiled within microscopic nuclei. Every body that moved, fed, or defended itself was guided by instructions carried in strands of deoxyribonucleic acid, instructions that determined where a head would form, how many segments would follow, and which tissues would differentiate into muscle, nerve, or gut. During this interval, regulatory systems that had long existed in simpler contexts began to orchestrate more elaborate forms. Among the most influential of these systems were the Hox genes, clusters of regulatory sequences arranged along chromosomes in a specific order. Their position within the genome corresponded to the spatial layout of the body, such that genes located at one end of the cluster influenced anterior structures, while those at the opposite end governed posterior regions. This collinearity allowed a developing embryo to establish a coordinated axis from front to back, assigning identity to successive segments. A slight shift in the timing or intensity of Hox gene expression could alter the number of segments or modify the appendages that emerged from them. Such changes need not be dramatic to produce visible differences. A mutation that extended the activation of a regulatory gene by a small interval could generate an additional body segment, providing new surfaces for muscle attachment or sensory placement. Another mutation might reposition a limb along the axis, creating novel patterns of movement. Because these genes acted high within the developmental hierarchy, their effects cascaded through networks of downstream targets, amplifying modest genetic variation into structural diversity. This developmental flexibility offered evolutionary advantage during a period of ecological expansion. As interactions intensified and habitats diversified, bodies capable of subtle modification could explore new niches without requiring entirely new genetic material. Existing pathways were repurposed, recombined, and fine tuned. The genome functioned less as a static blueprint and more as a dynamic toolkit, with regulatory elements adjusting growth patterns in response to selection pressures operating across generations. Evidence for this genetic groundwork comes not only from fossils, but from comparative genomics in living animals. Modern arthropods, vertebrates, and mollusks retain Hox clusters whose organization reflects deep ancestral origins. The shared architecture of these genes suggests that the potential for complex body plans had accumulated well before it became outwardly visible in mineralized form. What changed during the Cambrian was not the sudden invention of genetic machinery, but the context in which it operated. Environmental thresholds, ecological interactions, and rising oxygen levels created conditions under which latent developmental possibilities could be expressed and stabilized. In this sense, the genetic unlock was both ancient and immediate. The instructions had been present, quietly conserved within lineages that left little trace in earlier rocks. When selection began favoring articulated bodies, directed movement, and specialized organs, those instructions provided the means. From coordinated segmentation to patterned appendages, complexity unfolded from regulatory sequences that had waited, poised within cells, until the world around them invited their expression. Far below the impressions of shells and the branching of early limbs, the rocks themselves carry a subtler record, one written in ratios of isotopes and the chemistry of ancient minerals. Within carbonate layers and iron-rich sediments, geochemists trace shifts in oxidation states that speak of a gradual but decisive increase in available oxygen. Sulfur isotopes fractionate differently when oxygen penetrates deeper into marine environments. Redox-sensitive elements, such as molybdenum and uranium, accumulate in patterns that suggest more widespread ventilation of seawater. These signals do not announce a sudden surge, but rather a crossing of a threshold where background levels became sufficient to sustain new metabolic demands. Oxygen functions as a potent electron acceptor in cellular respiration, enabling organisms to extract far more energy from organic molecules than anaerobic pathways allow. With higher yields of adenosine triphosphate, cells can support active transport, sustained contraction, and the maintenance of complex tissues. Muscles that propel bodies through water require continuous energy input. Neurons that coordinate movement depend on stable electrochemical gradients. As oxygen concentrations rose, the energetic ceiling lifted, permitting greater body size and more intricate internal organization. Diffusion alone can supply small, flattened organisms with sufficient oxygen as molecules move passively across short distances. Larger bodies, however, present a different challenge. As thickness increases, the interior becomes distant from the surrounding medium, and simple diffusion cannot meet demand. Elevated environmental oxygen reduces this constraint, allowing tissues to remain viable even as volume expands. In time, circulatory systems would evolve to distribute gases efficiently, but the initial expansion in size required that the surrounding water contain enough dissolved oxygen to make such growth feasible. The interaction between biology and chemistry did not move in a single direction. As animals began burrowing into sediment, they disrupted previously stable layers, introducing oxygen into depths that had long remained anoxic. This process, sometimes called bioturbation, enhanced mixing between water and substrate, altering microbial communities and accelerating nutrient cycling. Oxygen penetrated farther below the seafloor, changing the balance of chemical reactions and influencing the availability of elements essential for life. In turn, these altered conditions could support additional metabolic activity, reinforcing the trend toward more active ecosystems. Geochemical records indicate that by the early Cambrian, oxygen levels in shallow marine environments approached a range capable of sustaining complex multicellular organisms. The atmosphere itself had been accumulating oxygen for hundreds of millions of years due to photosynthetic microbes, yet only when concentrations crossed a functional threshold did the energetic possibilities expand fully. Bodies could become thicker, more muscular, and more responsive without suffocating their inner cells. Thus, the rise of oxygen represents not a solitary cause, but a necessary backdrop against which anatomical and ecological innovations could persist. It set the stage upon which genetic potential and behavioral complexity could unfold, ensuring that once organisms began to move, hunt, and burrow, the chemistry of the ocean would not constrain them back into simplicity. Across Cambrian seafloors, the surface that had once lain smooth and continuous began to fray. Sediment layers that for immense stretches of time had rested undisturbed beneath thin films of microbial life were pierced, folded, and reworked from within. Narrow tunnels appear in cross-section through ancient rock, sinuous traces that record the passage of bodies moving beneath the surface rather than across it. The boundary between water and substrate, once stable and laminated, became porous and alive with motion. These early burrowers did not simply travel. They transformed their environment as they moved. By pushing through sediment in search of organic matter or refuge, they disrupted the cohesive microbial mats that had long stabilized the seabed. Those mats, once continuous carpets that limited exchange between overlying water and underlying layers, were broken into fragments. Oxygenated water began to penetrate more deeply into the sediment column, carried downward through freshly opened pathways. The chemistry of the seabed shifted as reduced compounds met oxygen and underwent new reactions. This process, now termed bioturbation, altered the distribution of nutrients and trace elements within marine ecosystems. Phosphorus, nitrogen, and iron cycled differently when sediment was mixed rather than stratified. Microbial communities adapted to these changing conditions, some retreating to deeper anoxic zones, others expanding into newly oxygenated niches. The physical act of digging thus initiated a cascade of chemical and biological consequences that extended beyond the individual animal. The structure of habitats changed as well. Instead of a uniform microbial surface, the seabed became a three-dimensional mosaic of tunnels, chambers, and disturbed patches. Some organisms specialized in grazing along freshly exposed surfaces. Others exploited the protective spaces within burrows. Predators could follow prey into sediment or lie in wait at tunnel openings. The very architecture of the ecosystem gained depth, adding vertical complexity to what had once been largely horizontal communities. Trace fossils from this interval reveal increasing diversity in burrow shapes and patterns. Simple horizontal tubes give way to branching networks and vertical shafts, suggesting experimentation in feeding strategies and modes of life. The sediment itself became a record not only of body form but of behavior, preserving the pathways through which animals interacted with their surroundings. Each new trace indicates that the substrate was no longer merely a surface to rest upon, but a medium to inhabit and reshape. As mixing intensified, The feedback between biology and chemistry strengthened. Oxygen, drawn into sediment, supported aerobic microbes that decomposed organic matter more efficiently, releasing nutrients back into the water column. Enhanced nutrient availability could in turn support greater primary productivity above, sustaining larger and more active animal populations. The system reorganized around this new dynamic, one in which the actions of burrowing organisms continuously modified their own habitat. From this point onward, the ocean floor would never return to its former stillness. The laminar calm of earlier ages gave way to a churned landscape, structured by movement and maintained through constant interaction. In reshaping sediment, early animals reshaped the chemical and ecological fabric of the sea, embedding their influence not only in fossils of their bodies, but in the very texture of the rock beneath them. High in the Canadian Rockies, where steep ridges rise above alpine forests and thin air sharpens the horizon, layers of dark shale rest quietly between bands of limestone. In the summer of 1909, paleontologist Charles Doolittle Walcott paused along one such slope and noticed slabs that split with unusual smoothness, revealing faint silhouettes pressed into stone. What he uncovered would become known as the Burgess Shale, a deposit unlike almost any previously studied, preserving not only shells and hard fragments, but the delicate outlines of soft tissues. These rocks had formed in an ancient marine basin where fine sediment settled rapidly and, under specific chemical conditions, limited decay. Bodies that would normally dissolve or be consumed were entombed before disintegration could erase their details. Muscle fibers, digestive tracts, and even traces of sensory structures appear as dark films against lighter stone. The preservation is so precise that anatomical relationships can be reconstructed with confidence, revealing entire organisms that lack mineralized skeletons. When these fossils were first described, many seemed unfamiliar, their body plans not easily assigned to recognized groups. Spiny lobopodians, segmented swimmers, and creatures bearing unusual appendages emerged from the shale in astonishing variety. The assemblage captured a snapshot of Cambrian ecosystems with a completeness rarely seen elsewhere, displaying both common species and rare forms that left few descendants. It was as though a curtain had been lifted, allowing scientists to view a community in motion rather than a collection of isolated fragments. For decades, interpretations were shaped by prevailing frameworks that favored fitting new discoveries into established categories. Some Burgess Shale organisms were reconstructed in ways that later proved inaccurate, their orientations or affiliations misunderstood. As additional specimens were collected and analytical methods improved, revisions followed. The diversity documented within these layers suggested that early animal evolution involved a broader range of anatomical experimentation than previously assumed. The impact on scientific thought was gradual but profound. Instead of viewing the Cambrian interval as a simple progression toward modern forms, researchers began to appreciate its complexity and contingency. The presence of entire lineages that flourished briefly and then disappeared highlighted the selective nature of evolutionary survival. Soft-bodied taxa, once largely invisible in the fossil record, gained recognition as integral components of early ecosystems. The Burgess Shale demonstrated that preservation bias had concealed much of the true diversity of ancient life. Where typical deposits favored hard parts, this exceptional site revealed the fragile architectures that underpinned ecological networks. By expanding the range of observable anatomy, it reshaped discussions about the tempo and mode of early animal diversification. The discovery did not introduce new life into the Cambrian seas. It illuminated what had long been there, recorded in shale that waited patiently for careful hands to split it open and read its quiet testimony. On another continent, far from the steep slopes of the Canadian Rockies, fine-grained mud once settled quietly along the margins of a Cambrian sea that covered what is now southern China. Within those sediments, later uplifted and exposed in Yunnan province, lies a deposit known as the Chengjiang Biota, a window into life that predates many of the forms described from North America. When these fossils were brought to light in the late 20th century, they extended the record of complex animals deeper into the early Cambrian, revealing communities already rich in structure and diversity. Like the shale in Canada, the Chengjiang deposits preserve soft tissues with remarkable fidelity. Bodies appear flattened yet detailed, their internal organs outlined in subtle contrasts of mineral replacement. Delicate appendages, gill filaments, and digestive tracts are visible where decay was halted by rapid burial and low oxygen conditions at the seafloor. The assemblage includes arthropods, worms, and a range of enigmatic organisms, many of which display anatomical complexity comparable to later Cambrian faunas. Among the most significant discoveries are early chordates, small, elongated animals bearing a notochord that runs along the length of the body. This flexible rod, composed of specialized cells, provided axial support and foreshadowed the vertebral columns that would arise in distant descendants. Specimens such as Hycoithus and Miloconmingia show evidence of segmented musculature and structures interpreted as primitive gill regions. Though modest in size, these animals represent foundational steps in the lineage that would eventually include fish, amphibians, reptiles, mammals, and humans. The presence of such organisms in the Chengjiang biota indicates that key body plans had already diversified early in the Cambrian period. Rather than a sudden appearance confined to a narrow interval, the fossil record suggests a more extended unfolding, with multiple lineages established within a relatively brief span of geological time. Radiometric dating of associated volcanic layers places these deposits several million years earlier than the classic Burgess Shale assemblage, implying that the diversification of complex animals was underway sooner than previously recognized. This deeper timeline does not diminish the intensity of the Cambrian transition, but it refines its contours. Genetic and ecological innovations likely accumulated incrementally before becoming conspicuous in the fossil record. The Chengjiang fossils capture a phase when many of the fundamental architectures of animal life were already present, interacting within dynamic ecosystems shaped by movement, predation, and environmental feedback. By expanding the geographic and temporal scope of early animal fossils, the discoveries in China underscore that the rise of complex life was not confined to a single basin or locality. It was a global phenomenon, expressed in different marine settings across ancient continents. The sedimentary rocks of Yunnan preserve this earlier chapter with clarity, demonstrating that the roots of modern body plans extend slightly further back into deep time than once assumed, woven into marine communities that thrived beneath Cambrian waters long before their story was fully understood. In the faint impressions of slender bodies preserved within early Cambrian mudstone, a subtle line runs from head to tail, barely visible yet profoundly significant. This line is not armor, not a shell, but a flexible rod of cells arranged in a precise column, a notochord that provided internal support without rigidity. In small chordates, such as Hycoichthys, this structure formed the central axis around which muscle blocks were organized, giving the body both stability and fluid motion through water. Unlike external skeletons that encased soft tissue, the notochord lay within, embedded beneath layers of developing muscle. It functioned as a hydrostatic scaffold, resisting compression while allowing lateral bending. As segmented muscles contracted along alternating sides of the body, the notochord stored and released elastic energy, producing smooth undulations that propelled the animal forward. Movement became coordinated along a single axis, guided by neural tissue that extended above the notochord as a primitive dorsal nerve cord. This internal framework marked a departure from earlier body plans. Instead of relying solely on external armor or diffuse support from surrounding water, these organisms carried their own structural core. The arrangement permitted greater precision in locomotion and directional control, qualities that would become essential in more complex environments shaped by active predation and competition. Gill structures along the sides of the body suggest that these early chordates were already extracting oxygen efficiently as they swam, integrating respiration with movement. The notochord also established a developmental pattern that would echo through deep time. During embryonic growth, it acted as a signaling center, releasing molecular cues that influenced the formation of surrounding tissues. Its presence helped organize the neural tube above and the somites on either side, ensuring that muscles and nerves aligned correctly along the body axis. In later vertebrates, the notochord would be partially or wholly replaced by a vertebral column composed of bone or cartilage, yet its foundational role remained embedded in developmental pathways. Fossils of Hycoichthys and related forms are modest in scale, yet they represent a pivotal stage in the evolution of the vertebrate lineage. The segmentation of musculature, the arrangement of sensory structures near the head, and the internal support provided by the notochord together form a blueprint that underlies fish, amphibians, reptiles, birds, and mammals. Though these early chordates lacked jaws and complex skeletons, their anatomy contains the essential architecture upon which later diversity would build. Within Cambrian seas, such organisms were only one component of a broader community, yet their design carried implications far beyond their immediate surroundings. The establishment of an internal axial support system enabled new possibilities for size, speed, and structural refinement. From this quiet beginning, preserved in stone as a thin line through an ancient body, the lineage that would eventually stand upright on land first traced its outline beneath shallow marine waters. When layers of rock are stacked one upon another across immense spans of time, they do not preserve every moment equally. Some intervals leave behind abundant traces, while others pass with little more than faint chemical signatures. The apparent abruptness of early animal diversification emerges in part from this uneven archive. For hundreds of millions of years, organisms lacking mineralized tissues lived, reproduced, and evolved in ways that rarely translated into durable fossils. Soft bodies collapse quickly after death, their forms erased unless burial is rapid and conditions are precise. As skeletons and hard coverings became more common, the likelihood of preservation increased dramatically. Mineralized shells resist decay, survive compaction, and remain visible long after surrounding sediment has turned to stone. This shift alone amplifies the fossil record, making it seem as though complexity appears suddenly. In reality, earlier lineages may have been diversifying quietly, their genetic and anatomical experiments largely invisible to future observers. The rock record captures a threshold where biological innovation and preservational opportunity intersect. Geological time further complicates perception. 20 million years, though brief compared with the billions that preceded it, encompasses spans far longer than human history. Within such an interval, countless generations can accumulate incremental change. A developmental pathway may be refined gradually, mutations filtered by selection across millions of reproductive cycles. When viewed through the compressed lens of stratigraphy, these cumulative steps can appear as a rapid burst, yet they likely represent the culmination of extended preparation. Genetic systems capable of supporting complex bodies did not materialize instantaneously. Regulatory networks evolved through earlier phases, establishing patterns of segmentation, polarity, and tissue differentiation. The eventual expression of these capacities during the Cambrian reflects not spontaneous invention, but the convergence of prior groundwork with favorable environmental conditions. Oxygen levels had risen sufficiently, ecological interactions had intensified, and developmental flexibility allowed latent potential to be expressed in visible form. Radiometric dating and careful correlation between global stratigraphic sections refine this timeline, revealing that diversification unfolded over millions of years rather than in a single moment. Even within Cambrian strata, the appearance of different groups is staggered, with some lineages emerging earlier and others later. The perception of suddenness arises because these events are clustered within a relatively narrow window when compared to the deep stretches of simpler life that preceded them. Understanding this perspective softens the sense of abrupt transformation without diminishing its significance. The Cambrian interval represents a genuine acceleration in ecological and anatomical complexity, yet it rests upon foundations laid long before mineralized remains began to accumulate in abundance. The seeming explosion is, in part, an artifact of preservation and scale, a reminder that Earth's history is filtered through layers that favor certain stories over others. Through careful study of both fossils and geochemistry, a more continuous narrative emerges, one in which preparation and expression are woven together across deep time. Along Cambrian coastlines and within open marine shelves, interactions that had once been tentative settled into a persistent exchange of pressure and response. Each successful strike by a predator altered the balance of survival. Each narrow escape left behind individuals whose traits conferred slight advantage. Over generations, these marginal differences accumulated, shaping bodies in ways that reflected ongoing contest rather than static design. The ecological fabric grew denser, threaded with strategies that met and countered one another in steady rhythm. Predators refined their capacity for capture. Appendages curved more effectively around struggling forms. Grasping surfaces developed ridges and denticles that improved grip. Musculature strengthened, enabling bursts of acceleration that reduced the margin for evasion. Sensory systems integrated information more efficiently, coordinating movement with detection. In response, prey species altered their own architecture. Spines lengthened and multiplied, projecting outward to complicate swallowing or penetration. Exoskeletons thickened, layering mineral deposits into patterns that resisted fracture. Body shapes flattened or streamlined, minimizing exposed surfaces and enhancing maneuverability. Speed became an asset not only for hunters but for those pursued. Streamlined profiles reduced drag, while segmented musculature allowed sustained undulation through water. The capacity to turn sharply or dive into sediment offered momentary refuge. Even coloration and surface texture likely played roles, though these are rarely preserved in stone. Beneath these visible adaptations lay genetic variation continuously sorted by selection, favoring combinations that enhanced survival within a shifting network of threats and opportunities. This reciprocal dynamic produced feedback that amplified evolutionary change. As defenses improved, predatory tactics intensified. As predators gained efficiency, prey responded with new layers of protection or novel behaviors. The pace of modification need not have been frantic to be transformative. Over spans of millions of years, incremental adjustments compounded into substantial divergence from ancestral forms. Natural selection, operating without foresight, nonetheless generated a landscape of escalating complexity. Such escalation did not remain confined to a few lineages. It permeated communities, influencing organisms indirectly connected within food webs. Herbivores grazing upon microbial films altered nutrient cycles, affecting the abundance of prey available to carnivores. Apex hunters shaped the distribution of mid-level consumers, which in turn influenced lower trophic levels. Evolution thus fed upon itself, with each adaptation altering selective pressures experienced by others. The arms race that began in Cambrian seas did not conclude within that period. Its logic persists in modern oceans, forests, and grasslands, wherever organisms interact in ways that influence survival and reproduction. The early establishment of active predation set in motion a self-reinforcing cycle, one in which innovation begets counter-innovation. From that foundation, the diversity of animal life expanded, structured not by a single event, but by continuous exchange, etched into anatomy through countless encounters beneath ancient waters. Across the Cambrian world, the continents lay arranged in configurations unfamiliar to modern maps, their edges low and often submerged beneath expansive inland seas. The climate during this interval was broadly warm, shaped by elevated concentrations of atmospheric carbon dioxide and the absence of extensive polar ice. Without large ice sheets to lock away water, global sea levels remained high, flooding continental margins and creating vast tracts of shallow marine habitat bathed in sunlight. These epiric seas, spread thinly across continental interiors, were neither deep nor turbulent in most regions. Sunlight penetrated easily to the seafloor, sustaining photosynthetic organisms that formed the base of marine food webs. The water column, warmed by greenhouse conditions, experienced limited temperature gradients compared with later periods marked by stronger latitudinal contrasts. Such relative thermal stability reduced seasonal extremes, allowing ecosystems to develop with fewer abrupt climatic disruptions. Shallow environments offer structural complexity that deeper basins lack. Variations in substrate, from fine mud to carbonate sand, created microhabitats that different organisms could exploit. Gentle currents transported nutrients while avoiding the destructive force of frequent storms. In these conditions, early animal communities found room to diversify. Grazers moved across illuminated bottoms. Burrowers reworked sediment beneath. Swimmers traversed clear water above. The combination of warmth, light, and nutrient availability supported high biological productivity. Geochemical evidence indicates that the Cambrian atmosphere maintained sufficient greenhouse gases to prevent large-scale glaciation. Carbon isotope records from marine carbonates suggest active carbon cycling, influenced by both volcanic outgassing and biological sequestration. Weathering of exposed land surfaces delivered ions to the oceans, contributing to carbonate precipitation and influencing seawater chemistry. This interplay between tectonics, atmosphere, and ocean established a climatic backdrop that favored marine expansion. The high sea levels also increased the length of coastlines relative to land area, multiplying ecological boundaries where shallow water met emerging continents. Such margins are often sites of intensified biological interaction as nutrients from land mix with marine currents. The proliferation of these transitional zones likely enhanced opportunities for colonization and adaptation, fostering the spread of diverse animal lineages. While regional variations certainly existed, the overall stability of Cambrian climate reduced barriers to dispersal across shallow seas. Marine corridors connected different basins, allowing organisms to expand their ranges and encounter new competitors or prey. This connectivity supported the global distribution of many early animal groups, reflected in fossil assemblages found on multiple paleocontinents. The Cambrian climate did not dictate the specific forms that life assumed, yet it provided environmental continuity against which evolutionary processes could operate effectively. Warm waters, ample light, and extensive shallow habitats created a setting in which genetic potential, ecological interaction, and chemical conditions converged. Within these greenhouse seas, the architecture of modern animal ecosystems took root, shaped by climate that remained permissive rather than restrictive during this formative chapter of Earth's history. Far beneath the seas where early animals swam and burrowed, the solid crust of the planet was in motion, shifting slowly along boundaries that would only much later be understood. The land masses of the late Precambrian had once been joined in larger assemblies, but by the early Cambrian, these supercontinents were fragmenting. Rifts opened, plates diverged, and new ocean basins widened between separating blocks of crust. The rearrangement unfolded at a pace imperceptible within a single lifetime, yet across millions of years it reshaped the geography upon which marine ecosystems depended. As continents broke apart, their margins multiplied. Each new coastline formed a transition between land and shallow sea, increasing the total length of continental shelves exposed to sunlight. These submerged platforms, gently sloping and rich in sediments derived from weathering rock, became expansive habitats for marine life. Shallow water environments differ profoundly from deep ocean basins. They receive greater light, experience more nutrient input from rivers, and provide varied substrates upon which organisms can anchor, graze, or hunt. The fragmentation of larger landmasses also altered ocean circulation. Currents flowed differently around isolated continental blocks, redistributing heat and nutrients in patterns that influenced local climates and productivity. Regions once connected became separated by expanding seas, allowing populations of organisms to diverge in relative isolation. Genetic differences accumulated as communities adapted to slightly distinct conditions of temperature, salinity, and substrate type. Over time, such geographic separation fostered the emergence of new lineages. Sedimentary records from multiple paleocontinents reveal both shared and regionally unique fossil assemblages, reflecting this balance between connectivity and isolation. Some animal groups achieved wide distribution, carried by currents across broad marine corridors. Others remained restricted to particular basins, their anatomical traits shaped by local ecological pressures. Geography thus became an active participant in evolutionary trajectories, not merely a passive backdrop. The creation of additional coastlines expanded the diversity of ecological niches available. Rocky substrates along some margins supported encrusting organisms and grazers, while muddy deltas favored burrowers and deposit feeders. Carbonate platforms developed in warm, clear waters, providing settings for skeletal organisms to accumulate and build layered deposits. Variations in depth, energy levels, and sediment supply produced mosaics of habitat that encouraged specialization. Species could exploit narrow ranges of conditions, refining their anatomy and behavior in response to specific environmental cues. Over geological timescales, plate movements continued to rearrange continents, altering proximity and opening or closing marine passages. These shifts influenced dispersal pathways and competitive interactions, sometimes bringing previously separated faunas into contact. Biodiversity patterns visible in the Cambrian fossil record reflect this dynamic geography shaped by tectonic processes operating far below the water's surface. Through the slow drift of continents and the expansion of shallow seas, the physical stage for early animal life became increasingly intricate. Coastlines lengthened, habitats diversified, and barriers both formed and dissolved. In this interplay between stone and water, biodiversity found space to expand, guided not only by biology and chemistry, but by the restless movement of the planet itself. Not every lineage that once lay upon ancient seafloors continued into the Cambrian world. In strata that predate the rise of active predation and mineralized armor, impressions of quilted forms and frond-like bodies appear with quiet regularity. Then, as layers shift upward through time, many of those distinctive shapes grow scarce and finally absent. The sediment records a transition in community composition, a turnover that suggests more than simple addition of new forms. Ediacaran organisms, often soft-bodied and lacking clear evidence of mouths or complex internal organs, had occupied ecological space suited to relatively stable substrates. Their growth patterns imply absorption of nutrients across body surfaces, and their broad contact with sediment indicates intimate association with microbial communities. When burrowing intensified and sediments were mixed more deeply, the surfaces upon which these organisms depended changed character. Cohesive microbial layers fragmented, and oxygen penetrated previously undisturbed zones, altering chemical gradients that may have supported earlier life strategies. At the same time, the emergence of mobile grazers and predators introduced new pressures. Bodies resting openly upon the seafloor faced risks they had not encountered before. Defensive structures were largely absent among Ediacaran taxa, and their modes of feeding may not have been compatible with increasingly dynamic environments. Some lineages might have adapted or persisted in restricted habitats, yet many leave no clear descendants in younger rocks. Whether these disappearances represent true extinction or gradual replacement remains a subject of ongoing study. Fossil preservation biases complicate interpretation since soft-bodied organisms are less likely to be recorded once sedimentary conditions change. It is possible that certain Ediacaran forms evolved into more derived animals whose ancestry is obscured by morphological transformation. In other cases, ecological competition and predation may have driven populations beyond recovery, reducing their numbers until they no longer appear in the geological record. What is evident is that the structure of marine communities shifted markedly during this interval. The relative abundance of quilted and frond-like impressions declines as trace fossils and mineralized remains become more common. Energy pathways through ecosystems reorganize, reflecting new interactions and altered resource use. The turnover is not necessarily catastrophic in the sense of sudden global collapse. Rather, it appears as a sustained reconfiguration in which some forms diminish while others expand. This pattern underscores that the Cambrian transition was not solely an episode of creative diversification, but also one of selective loss. As environmental conditions, chemistry, and biological interactions changed, certain body plans proved less suited to the emerging landscape. Their absence in later strata marks the closing of an earlier chapter in Earth's history, one characterized by different modes of living. In their place, more mobile and structurally complex organisms established dominance, reshaping the seafloor and redefining the possibilities of animal life. Scattered through Cambrian strata are forms that seem to flicker briefly in the record and then recede, their outlines preserved in exquisite detail, yet lacking continuation in later periods. These organisms possessed combinations of segments, appendages, and protective structures that do not map cleanly onto enduring lineages. They appear as if drawn from a broader range of anatomical possibilities than those that ultimately persisted, each design representing a pathway explored and then set aside. Some bore elaborate arrays of spines without corresponding increases in mobility. Others combined feeding structures and configurations that proved difficult to sustain as ecosystems intensified. Certain body plans relied heavily on specific environmental conditions, thriving in localized settings but failing to expand when geography and climate shifted. The Cambrian seas were laboratories of form where developmental systems, newly flexible and responsive, generated a spectrum of architectures that extended beyond what later ages would retain. Natural selection acted not as a deliberate sculptor, but as a steady filter. Traits that enhanced survival and reproduction within changing communities were propagated. Those that conferred only temporary advantage diminished over time. This filtering process did not eliminate variation entirely, yet it narrowed the range of viable structural blueprints. Designs that could integrate feeding efficiency, defense, locomotion, and reproduction within a coherent framework tended to endure. Others, though ingenious in isolation, lacked the balance required to navigate long-term ecological pressures. The fossil record preserves evidence of these vanished experiments in deposits where soft tissues and delicate appendages were captured before decay erased them. In younger rocks, their absence speaks quietly of extinction rather than transformation. Evolution is not solely a story of accumulation. It is equally a chronicle of loss. Each lineage that disappeared carried unique genetic combinations, developmental pathways, and ecological roles that ceased to influence subsequent history. Over millions of years, as climates fluctuated, continents drifted, and interactions intensified, this filtering reshaped marine communities. Certain foundational body plans, such as those underlying arthropods, mollusks, and chordates, demonstrated resilience across diverse environments. Their modular organization and capacity for incremental modification allowed adaptation without wholesale redesign. In contrast, more rigid or highly specialized forms proved less adaptable when circumstances shifted. The notion of experimentation in early animal evolution does not imply randomness without constraint. Developmental biology channels variation along permissible axes, and environmental context further limits outcomes. Yet within these boundaries, Cambrian ecosystems supported an extraordinary diversity of anatomical expression. The long-term persistence of only a subset of these forms reflects both the opportunities and limits inherent in biological innovation. Through deep time, this process of testing and filtering continued, shaping the distribution of life on Earth. The Cambrian interval captures an especially vivid phase of exploration when many structural possibilities were trialed in rapid succession. What remains in later periods represents not the totality of what once existed, but the residue of countless variations refined by selection across vast spans of geological history. By the close of the Cambrian, the broad outlines of animal design had settled into recognizable frameworks, not in their final forms, but in structural blueprints that would persist through all subsequent ages. The fundamental arrangements of tissues, symmetry, segmentation, and internal organization that define major phyla were already present in marine communities. Arthropods carried jointed appendages organized around segmented bodies. Mollusks displayed muscular feet and mantle cavities. Chordates bore axial supports and dorsal nerve cords. These architectures established the primary ways in which complex, multicellular life could be assembled. What followed across later periods was not the invention of entirely new structural themes, but the refinement and diversification of those already in place. Within each phylum, variation expanded dramatically. Arthropods radiated into forms that crawled across land, swam in oceans, and eventually took to the air. Yet their exoskeletons, segmented bodies, and jointed limbs remained consistent features. Chordates diversified into jawed fish, tetrapods, birds, and mammals, yet the underlying axial plan, bilateral symmetry, and pattern musculature trace back to early templates. Developmental biology helps explain this continuity. The genetic regulatory networks that govern body layout operate early in embryogenesis, establishing axes and segment identities that cascade into later structures. Alterations to these foundational systems can have widespread and often deleterious consequences, limiting the range of viable modifications. Evolution, therefore, tends to adjust peripheral features such as limb length, ornamentation, or feeding apparatus, while conserving core organizational patterns that maintain functional integrity. The fossil record reflects this pattern of stability within diversity. Cambrian strata reveal the initial establishment of major phyla, and subsequent layers show elaboration within those groups rather than replacement by wholly novel body architectures. Extinctions occurred and entire clades vanished, yet the basic phylum-level designs endured. They proved adaptable enough to withstand climatic fluctuations, continental rearrangements, and mass extinction events that reshaped the biosphere multiple times. This persistence does not imply stagnation. On the contrary, evolutionary innovation continued vigorously, generating new species, ecological roles, and complex behaviors. Feathers evolved from reptilian scales, mammalian jaws transformed from ancestral gill arches, and insect wings arose from modified body segments. Each innovation built upon pre-existing structural frameworks, demonstrating the capacity of established architectures to accommodate remarkable diversity without abandoning their foundational patterns. The Cambrian interval thus represents a moment when the primary construction plans for animal life crystallized. Once these frameworks demonstrated their versatility and resilience, natural selection operated within them, exploring permutations and refinements rather than seeking entirely different blueprints. Through deep time, the outward expressions of life have shifted repeatedly, yet beneath those changes, the same essential designs persist, linking modern organisms to their ancient marine ancestors in a continuous structural lineage that began in those formative seas. Measured against the span of human memory, 20 million years is incomprehensible, a duration so vast that language falters when attempting to hold it. Yet within the chronology of Earth, that interval occupies only a thin layer in the stratigraphic column, a narrow band of sediment resting atop billions of years that preceded it. When geologists describe the Cambrian diversification as rapid, they do so not by everyday standards, but by comparison with the immensity of deep time. For more than 3 billion years before the Cambrian, life remained primarily microscopic, its innovations unfolding slowly across planetary scales. Entire mountain ranges rose and eroded, oceans opened and closed, atmospheric chemistry shifted from oxygen poor to oxygen rich. Through all of this, multicellular complexity remained limited, and ecosystems were structured around simple interactions. The temporal ratio is striking. The preparatory phase spanned billions of years, while the establishment of major animal body plans occurred within tens of millions. In a geological context, 20 million years can be visualized as a modest slice within a much thicker sequence of rock. Sedimentary layers representing far longer intervals may compress into similar thickness depending on deposition rates. Radiometric dating techniques, relying on the decay of isotopes such as uranium into lead, allow scientists to assign ages with increasing precision. When these ages are plotted against fossil occurrences, the clustering of key anatomical innovations within a relatively confined window becomes apparent. The designation of rapid does not imply instantaneous transformation. Evolution operates through successive generations, and even modest rates of genetic change can accumulate substantially over millions of years. However, when such accumulation is contrasted with the preceding billions, during which comparable levels of structural diversification did not occur, the difference in tempo becomes clear. The Cambrian interval represents an acceleration relative to background rates observed earlier in Earth's history. Geological comparison further emphasizes this point. The formation and breakup of supercontinents often span hundreds of millions of years. Major shifts in atmospheric composition can require similar durations. Against these processes, the widespread appearance of mineralized skeletons, complex sensory organs, and articulated body plans within a few tens of millions of years stands out as concentrated in time. It is a compression of biological innovation into a fraction of the planet's lifespan. Understanding this scale reshapes perception. What feels immense from a human perspective becomes comparatively brief when situated within 4.5 billion years of planetary evolution. The Cambrian diversification did not occur in a moment, yet within the cadence of geological time, it unfolded swiftly enough to leave a distinct boundary in the fossil record. That contrast between vast preparation and concentrated expression is why scientists describe those 20 million years as rapid, a relative measure grounded in the long, unhurried history that came before. Within the broad arc of Cambrian diversification, the record does not unfold as a steady upward curve. Instead, careful examination of stratigraphic sequences reveals intervals where diversity contracts before expanding again, where certain lineages diminish and others take their place. These fluctuations are not the sweeping mass extinctions that would later punctuate Earth's history, yet they mark episodes of ecological stress and adjustment that shaped marine communities in quieter ways. Sedimentary layers from different Cambrian basins preserve changes in fossil abundance and composition that correspond with shifts in environmental conditions. Variations in carbon isotope ratios indicate perturbations in the global carbon cycle, likely tied to changes in productivity, ocean circulation, or volcanic activity. Sea level oscillations, driven by tectonic processes and thermal expansion of seawater, altered the extent of shallow habitats. When sea levels fell, continental shelves contracted, compressing populations into reduced areas. When they rose, new habitats opened but required recolonization. Such environmental fluctuations influenced oxygen distribution in marine settings. Localized anoxic events, where dissolved oxygen declined in certain basins, could have stressed communities dependent on higher concentrations. Species with narrower tolerances may have declined, while others adapted to altered conditions persisted. These shifts are reflected in population turnovers visible in fossil assemblages, where once common taxa become rare and new forms rise in relative abundance. The instability inherent in early complex ecosystems also contributed to these turnovers. As predator prey dynamics intensified and new body plans proliferated, competitive interactions likely drove some lineages toward extinction even without dramatic environmental change. Ecological niches filled rapidly during the Cambrian, and when multiple species occupied similar roles, small advantages could determine survival over millions of years. Natural selection operated continuously, and the disappearance of certain groups may have resulted from gradual displacement rather than sudden catastrophe. These minor extinction events remind us that evolutionary radiations are rarely smooth expansions. Growth and loss occur together, intertwined across geological timescales. While the Cambrian period is often characterized by its creative energy, it also contained episodes of contraction that pruned diversity. Such pruning may have contributed to the stabilization of major body plans, filtering out lineages less suited to shifting conditions. Importantly, these fluctuations did not reverse the broader trajectory toward complex, structured ecosystems. After each downturn, diversification resumed, sometimes with new combinations of species and altered community structures. The fossil record thus captures a dynamic equilibrium where instability is woven into the fabric of early animal history. Through environmental variation and ecological competition, Cambrian seas experienced cycles of expansion and reduction, shaping the composition of life in ways that would influence subsequent periods long after these minor extinctions had passed into stone. As Cambrian seas gave way to the early Ordovician, the momentum of diversification did not fade. Instead, it broadened and settled into new patterns. The foundational body plans established earlier now supported expanding lineages that spread across continental shelves with increasing ecological complexity. Fossil assemblages from this interval reveal a steady rise in species richness, not as a sudden burst, but as sustained elaboration upon earlier frameworks. One of the most visible transformations during this transition was the expansion of reef-building communities. In Cambrian settings, reef structures were often modest, constructed primarily by microbial mats and simple skeletal organisms. By the Ordovician, reef systems grew larger and more architecturally intricate. Sponges, bryozoans, corals, and calcareous algae contributed to frameworks that rose above the seafloor, creating layered habitats within and around their rigid skeletons. These reefs altered water flow, trapped sediment, and provided shelter for diverse assemblages of invertebrates. The proliferation of such structures had cascading ecological effects. Elevated surfaces within reefs offered attachment sites for filter feeders that relied on steady currents. Crevices and cavities served as refuge for smaller organisms, while predators patrolled the outer margins. Energy flowed through these systems in increasingly complex pathways, linking primary producers, grazers, and carnivores in intricate food webs. The three-dimensional architecture of reefs added depth to marine environments that had once been comparatively flat. Marine dominance became more firmly established as terrestrial ecosystems remained limited in scope. While life persisted on land in microbial forms and perhaps simple plants, the oceans were the principal arenas of biological innovation. Brachiopods, trilobites, mollusks, echinoderms, and early vertebrates diversified across shallow seas that continued to cover broad continental areas. The expansion of planktonic organisms in the water column also contributed to increased productivity, supporting larger populations of suspension feeders and predators. Geological records indicate relatively stable climatic conditions during much of the early Ordovician, allowing marine communities to expand without abrupt global disruption. Continental positions and high sea levels maintained extensive shallow habitats similar to those that had fostered Cambrian diversification, yet the ecological tone shifted from experimentation toward consolidation. Many of the experimental body plans that characterized earlier phases were absent, replaced by lineages that demonstrated resilience and adaptability across varied settings. The transition to the Ordovician thus represents continuity rather than rupture. The groundwork laid in Cambrian seas matured into ecosystems that would endure, with modifications, for tens of millions of years. Reef frameworks expanded, species interactions deepened, and the dominance of marine life solidified as a defining feature of Earth's biosphere. The early chapters of complex animal evolution had given rise to communities capable of sustaining diversity at scales previously unseen, setting the stage for subsequent radiations that would carry life into new environments and forms. Beneath the surface of Ordovician seas, interactions that once unfolded in simpler patterns had matured into layered systems of exchange. Energy entered these waters through sunlight, captured by photosynthetic organisms, from microscopic phytoplankton suspended in the water column to algae attached to firm substrates. From that foundation, nourishment moved upward, transferred from one body to another in sequences that formed the early outlines of recognizable food chains. Yet even the term chain suggests a simplicity that no longer fully applied. Primary consumers grazed upon microbial films and drifting cells, converting plant matter into animal tissue. Suspension feeders strained particles from passing currents, their delicate structures tuned to the flow of water shaped by expanding reef frameworks. Predators targeted these consumers, selecting prey based on size, mobility, and vulnerability. At each step, energy diminished, lost as heat through metabolism, yet enough remained to sustain higher trophic levels. The efficiency of these transfers depended on both environmental conditions and the anatomical refinements achieved during earlier evolutionary phases. As species diversified, niche specialization intensified. Organisms occupied increasingly precise roles within communities, reducing direct competition by exploiting distinct resources or microhabitats. Some burrowed deeply into sediment, feeding on organic material mixed through the substrate. Others remained at the sediment surface, filtering or grazing. In reef environments, certain species inhabited sheltered cavities, while others colonized exposed ridges where currents were strongest. This partitioning of ecological space allowed greater numbers of species to coexist within the same geographic area. The structure of these communities formed complex webs rather than linear sequences. A single species might serve as prey for multiple predators and as consumer of several food sources. The removal or decline of one group could ripple outward, influencing populations beyond immediate interactions. Such interdependence increased resilience in some respects, as alternative pathways for energy flow could buffer localized disturbances. At the same time, it introduced sensitivity, since changes in one part of the web could propagate in unforeseen ways. The blueprint of modern ecosystems is rooted in these early configurations: trophic levels, nutrient cycling, and habitat stratification all trace their lineage to patterns established during the Cambrian and Ordovician transition. Chemical elements moved through biological systems with increasing complexity, linking organisms not only through direct encounters, but through shared participation in broader cycles. Carbon fixed by microscopic producers might pass through multiple consumers before returning to the water column through decomposition. In this developing architecture, the biosphere achieved a new degree of organization. Communities were no longer assemblages of loosely connected forms, but integrated networks structured by energy flow and specialization. Though later eras would introduce terrestrial forests, flying insects, and apex predators on land, the underlying logic of ecosystem dynamics had already taken shape beneath ancient seas. From these marine origins, the principles that govern contemporary ecological systems emerged, refined, but fundamentally continuous with their early expressions. If one could descend through modern ocean waters and then gently shift backward through time, the differences in species would be striking, yet the underlying patterns would feel familiar. In coral reefs, along continental shelves, and within open pelagic zones, predation continues to structure communities in ways that echo those early marine experiments. Fish pursue smaller fish with streamlined bodies and forward-focused eyes. Cephalopods extend flexible arms toward crustaceans. Crustaceans themselves grasp plankton, drifting in currents shaped by tides and temperature gradients. The identities have changed, yet the logic of pursuit and escape remains. Predation still shapes anatomy at every scale. Shells thicken in response to drilling gastropods. Spines extend from sea urchins to deter crushing jaws. Camouflage patterns disrupt outlines against shifting backgrounds of sand and reef. Speed and maneuverability are refined through muscle arrangement and skeletal support, whether in the swift flick of a tuna's tail or the sudden contraction of a squid's mantle cavity. These traits are not static inheritances, but products of ongoing selection, perpetually adjusted in response to evolving adversaries. Vision remains central to many of these interactions. The compound eyes of arthropods and the camera-style eyes of vertebrates trace their origins to early optical innovations. Modern fish rely on color discrimination and motion detection to locate prey and avoid predators. In clear tropical waters, coral reef species display intricate visual signals used for recognition, mating, and territorial defense. Even in deeper or murkier environments, light-sensitive systems adapt to available wavelengths, demonstrating that the translation of photons into information continues to guide behavior. The arms race that began in Cambrian seas has not concluded. It persists as a dynamic equilibrium, with each advance met by counter-adaptation. Predatory fish develop sharper teeth and stronger jaws. Prey species evolve thicker scales or behavioral strategies such as schooling that reduce individual risk. Invertebrates produce chemical defenses that deter consumption. Predators evolve resistance or alternative feeding strategies. These cycles operate across generations, sometimes accelerating in response to environmental change. Modern oceans also illustrate how complex ecological webs amplify these interactions. Apex predators influence the abundance and distribution of lower trophic levels, creating cascading effects that reshape entire ecosystems. The removal of large sharks from certain regions has altered the balance of reef communities, demonstrating how deeply predation is woven into marine structure. Energy continues to flow through layered networks, first assembled hundreds of millions of years ago. What began as experimental adjustments in ancient seas has matured into systems of remarkable resilience and intricacy. The foundational dynamics established during the Cambrian, active predation, visual perception, reciprocal adaptation, remain embedded in contemporary marine life. Modern oceans are not separate from that distant past. They are its continuation, animated by the same evolutionary dialogue that first transformed simple communities into interactive, responsive ecosystems. In the cells of every animal alive today, from the smallest marine invertebrate to the largest whale, there are sequences of nucleotides that carry echoes of an ancient beginning. These sequences do not announce themselves with visible drama, yet they encode regulatory patterns and developmental instructions first assembled during the earliest phases of complex animal evolution. The Cambrian seas have long receded, their sediments compressed into stone, but the genetic frameworks established there continue to operate within modern bodies. Every major animal lineage traces its ancestry back to that formative interval. The segmented arrangement of arthropods, the radial symmetry of echinoderms, the bilateral organization shared by vertebrates and countless invertebrates all reflect architectures consolidated more than 500 million years ago. Though later epochs introduced feathers, fur, antlers, and wings, these are elaborations layered upon older structural themes. Beneath variation in size, habitat, and behavior lies a common blueprint. Comparative genomics reveals this continuity with striking clarity. Genes that regulate body axes, segment identity, and organ placement are conserved across diverse taxa. Hox clusters in mammals mirror those found in insects and marine worms, arranged in sequences that correspond to anterior and posterior patterning. Proteins, guiding neural development, muscle formation, and sensory differentiation, often share deep homology, indicating descent from ancestral genes present in early chordates and other Cambrian organisms. The divergence of lineages modified expression patterns and regulatory interactions, yet the core elements persisted. This shared genetic heritage extends beyond visible anatomy. Cellular processes such as apoptosis, the controlled death of cells during development, and signal transduction pathways that mediate responses to environmental cues exhibit continuity across animal phyla. These mechanisms evolved within early multicellular contexts and proved versatile enough to support increasingly complex forms. As lineages branched and environments shifted, the same molecular tools were redeployed in novel combinations. The long shadow of the Cambrian is therefore not merely paleontological, but biological in an immediate sense. When a vertebrate embryo establishes its body axis, or when an arthropod molts its exoskeleton, it draws upon regulatory systems shaped in those ancient seas. Evolution has introduced refinements, duplications, and losses, yet it has rarely abandoned foundational patterns entirely. Deep continuity underlies apparent novelty. Recognizing this lineage reshapes perspective on modern biodiversity. The diversity visible today is immense, yet it is organized within boundaries set early in animal history. From coral reefs to terrestrial forests, organisms embody variations on themes first articulated when eyes opened in shallow water and skeletons hardened against predation. The Cambrian did not dictate every detail of subsequent evolution, but it established a structural vocabulary that remains in use. Across hundreds of millions of years, extinctions and radiations have altered the cast of characters inhabiting Earth. Still, the underlying grammar of animal life endures. carried in genes and body plans that trace their origins to that pivotal interval. The shadow of the Cambrian stretches forward through time, linking present forms to distant ancestors in a continuous evolutionary thread. Long after the ancient seas receded and their sediments hardened into stone, the story of early animal life continues to be assembled in laboratories and field sites scattered across the globe. The tools used today are precise, often microscopic, yet they reach backward across hundreds of millions of years, refining timelines and clarifying relationships that once seemed uncertain. Each method adds resolution to a chapter written in rock. Radiometric dating provides the temporal framework. Within layers of volcanic ash interbedded with sedimentary deposits, minerals such as zircon preserve isotopes of uranium that decay at predictable rates into lead. By measuring the ratio between parent and daughter isotopes, geochronologists calculate the age of the surrounding rock with remarkable accuracy. These measurements anchor fossil assemblages within specific intervals, allowing scientists to compare events across continents and reconstruct the sequence of diversification and turnover with increasing confidence. Fossil imaging has advanced beyond simple surface observation. High-resolution computed tomography scans reveal internal structures hidden within stone, capturing three-dimensional details without damaging fragile specimens. Synchrotron radiation, generated by particle accelerators, can detect subtle differences in mineral composition, outlining soft tissues that are otherwise invisible to the eye. Through these techniques, digestive tracts, neural tissues, and muscle arrangements emerge from compressed fossils, offering insight into anatomy once thought irretrievable. Developmental biology contributes another dimension by examining how living organisms build their bodies. By studying gene expression patterns in embryos of modern arthropods, mollusks, and vertebrates, researchers infer the regulatory networks that shaped early animal forms. Experimental manipulation of developmental genes demonstrates how small changes in timing or intensity can alter segmentation and appendage formation. These observations bridge the gap between ancient morphology and contemporary genetics, illuminating mechanisms that underlie the fossil record. Molecular clocks extend this perspective further into deep time. By comparing genetic differences among living species and calibrating them with fossil dates, scientists estimate when lineages diverged from common ancestors. Although uncertainties remain, such analyses suggest that some evolutionary splits occurred before their first appearance in preserved strata. Molecular data therefore complement paleontological evidence, revealing hidden branches of the evolutionary tree that left few direct traces in stone. Together, these approaches create a layered understanding. Radiometric dating situates events in time, imaging reconstructs form, developmental biology explains how structures arise, molecular clocks trace relationships beyond visible fossils. The study of early animal evolution is no longer confined to quarry and outcrop, but unfolds through collaboration between geology, physics, genetics, and computational analysis. Through these integrated methods, the distant Cambrian world becomes clearer, its innovations examined not only as relics of deep time, but as subjects of ongoing scientific discovery. Even after more than a century of excavation, imaging, and genetic analysis, the origins of complex animal life resist a single, definitive explanation. The broad contours of diversification are visible in rock and genome alike, yet the precise trigger that set events into motion remains debated. Evidence accumulates from multiple disciplines, and still the question of causation unfolds in layered complexity rather than clear resolution. One line of reasoning emphasizes the rise of oxygen as a necessary threshold. Geochemical proxies suggest that increasing atmospheric and oceanic oxygen concentrations preceded or coincided with the expansion of metabolically demanding bodies. Higher oxygen availability would have supported greater energy production, enabling thicker tissues, sustained movement, and more intricate organ systems. Yet oxygen alone does not explain why diversification clustered within a particular interval. Oxygen levels had risen in earlier periods without producing comparable anatomical variety, suggesting that additional factors were at work. Another perspective focuses on genetic readiness. Regulatory networks, such as those governed by Hox genes, were already in place before the Cambrian interval became conspicuous in the fossil record. Developmental systems capable of generating segmentation, polarity, and organ differentiation may have accumulated gradually, awaiting ecological circumstances that favored their expression. In this view, the diversification reflects the release of latent potential rather than the sudden invention of new genetic tools. Environmental thresholds provide further context. Changes in ocean chemistry, nutrient availability, and continental configuration may have created conditions that amplified evolutionary feedback. The disruption of microbial substrates, expansion of shallow habitats, and intensification of predator-prey interactions formed a network of influences rather than a single cause. Crossing certain ecological thresholds might have allowed positive feedback loops to accelerate diversification once it began. The interplay among these elements complicates efforts to isolate one decisive trigger. It is possible that oxygen levels, genetic architecture, and ecological restructuring converged within a narrow window, producing a synergy that none could achieve alone. Such convergence would explain both the relative rapidity of the event and the difficulty in attributing it to a singular factor. Ongoing discoveries continue to refine this picture. New fossil sites extend the temporal range of early animal forms. Improved dating techniques narrow chronological uncertainties. Genomic studies reveal deeper homologies and divergence times. Each finding adds nuance, sometimes supporting existing models, sometimes challenging them. The Cambrian transition emerges less as a solved problem than as an evolving dialogue between evidence and interpretation. What remains uncertain does not diminish what is known. Instead, it underscores the complexity of biological systems interacting with planetary processes. The story of early animal life is still being written, not only in stone, but in laboratories and field camps around the world. The debate over triggers persists, inviting careful inquiry rather than finality, and reminding us that even events hundreds of millions of years past can retain elements of mystery within the fabric of deep time. Stand at the edge of deep time, and the unfolding of complex animals can seem almost assured, as though life were steadily climbing toward intricacy with quiet determination. Yet the record beneath our feet suggests something far less certain. The emergence of coordinated bodies, active movement, and layered ecosystems depended on conditions that were neither permanent nor guaranteed. They arose within windows that could easily have narrowed or closed. For billions of years before animals diversified, life persisted in forms that required no skeletons, no rapid motion, no elaborate sensory systems. Microbial communities thrived within chemical gradients and soft substrates, sustained by metabolisms that functioned under limited oxygen and relatively stable ecological structures. Nothing in those early worlds demanded the rise of predators with grasping limbs or eyes tuned to shifting light. Complexity was possible, but not compelled. The eventual appearance of animals required the convergence of multiple thresholds. Oxygen concentrations had to reach levels capable of supporting energetically expensive tissues. Genetic regulatory systems needed sufficient flexibility to generate organized body plans. Marine environments had to provide stable yet dynamic habitats where ecological feedback could amplify innovation. Each factor depended on planetary processes that might have unfolded differently under altered tectonic patterns, volcanic outgassing rates, or climatic states. Contingency threads through every layer of this history. A prolonged episode of oceanic anoxia could have delayed or suppressed diversification. A shift in nutrient cycling might have favored microbial dominance for far longer. Even the configuration of continents influenced shallow seas and coastlines, shaping where ecological experimentation could occur. Earth's trajectory was shaped by interacting systems, not by a predetermined script. Evolution itself operates without foresight. Mutations arise without intention, and natural selection filters them through immediate conditions. The success of early animal lineages depended on local advantages that accumulated into broader transformations. Had environmental parameters differed slightly, alternative lineages might have dominated, or diversification might have remained modest. The forms familiar today were not inevitable outcomes, but survivors of a filtering process governed by circumstance as much as capability. Yet, within contingency, there is also resilience. Once ecological networks, incorporating predation, burrowing, and mineralized skeletons, became established, feedback loops reinforced their persistence. Energy flow intensified, niches multiplied, and developmental systems diversified further. The stabilization of these patterns created a new baseline from which later evolution proceeded, making reversal increasingly unlikely. The history of early animals, therefore, reflects both fragility and momentum. It emerged from a narrow alignment of atmospheric chemistry, genetic architecture, and environmental structure, and then built upon itself until it reshaped the planet's surface systems. In that balance between chance and constraint lies a reminder that Earth's present biosphere rests on conditions that once could have tipped another way, and that the continuity we observe today arose from pathways that were open only briefly within the vast span of geological time. There is a stillness that follows long contemplation, a moment when the movement of continents and currents seems to recede into something almost weightless. In that stillness, the realization settles gently. Complex life was not written into the fabric of Earth from the beginning. For most of the planet's history, the seas moved without eyes watching through them, without articulated limbs stirring their layers, without structured bodies casting shadows across the seafloor. Chemistry cycled, microbial communities endured. The world was alive, yet it was simple in form. For billions of years, no lineage crossed the threshold into coordinated tissues and mineralized frames. Genetic systems existed, replicating and adapting within narrow bounds. Oxygen accumulated and fluctuated. Continents assembled and separated. Through all of it, the emergence of animals remained a possibility rather than a certainty. The ocean held potential that was neither urgent nor inevitable. Then, within a comparatively brief interval of geological time, that potential expressed itself. Regulatory genes orchestrated new spatial patterns. Oxygen supported denser metabolisms. Ecological interactions intensified, reinforcing innovation. Bodies gained orientation and internal scaffolding. Predation and defense became woven into the structure of ecosystems. Energy began to flow through layered food webs with a complexity unseen in earlier ages. The transformation did not erase what came before, but it altered the trajectory of everything that followed. Sediments were mixed by burrowing organisms, reef systems expanded, shaping coastlines and nutrient cycles. Evolutionary pathways multiplied, each constrained by inherited architectures yet open to modification. From that point onward, the ocean was no longer a realm dominated solely by microbial films and drifting cells. It became a dynamic mosaic of interacting animals, each carrying genetic memories of a distant origin. This shift did not guarantee the eventual rise of any particular species. It did not promise intelligence or self awareness. It simply established a new baseline, a framework within which further diversification could unfold. The patterns set in motion during that early interval continue to reverberate through modern ecosystems. Every fin slicing through water, every shell forming along a shoreline, every sensory organ translating light into perception carries traces of that foundational change. To reflect on this is to sense the fragility embedded within continuity. The path from simplicity to complexity required conditions that aligned only briefly within deep time. Had those alignments faltered, Earth might have remained a world of microscopic persistence. Instead, the oceans reshaped themselves around new possibilities, and those possibilities endured. Now, in the quiet after tracing that arc, the immensity of time feels both distant and intimate. The sea still moves, as it always has, but it moves through structures born in that ancient interval. Beneath the surface, the legacy of that transformation remains active, subtle, and ongoing. Complex life was not guaranteed. For ages it did not appear. Then it did, and the planet has carried that imprint ever since.